Okay. Our second speaker this afternoon is Dr. Emily Coffey. Dr. Coffey earned her PhD from the University of Oxford, where she focused on island biogeography and the conservation ecology of rare plants. She holds leadership roles with organizations including Botanical Gardens Conservation International, the Global Conservation Consortium for Magnolia, and she co-founded the Southeastern Plant Conservation Alliance. At present, Dr. Coffey is the Vice President of Conservation and Research at the Atlanta Botanical Garden, where she leads efforts to protect biodiversity through cutting-edge conservation programs. Today, she'll speak on the crucial role of botanical gardens in plant conservation.
Good afternoon, everybody. It is so fantastic to see you all here today, and I'm incredibly honored and so grateful for this opportunity to speak to you today about a topic that is extremely near and dear to my heart. And I want to thank the Smithsonian and the U.S. Botanic Garden and the Smithsonian Gardens for the invitation. So it may seem very obvious to all of us. You're all here because you're interested in plants, but plants are really everything. They are absolutely essential for the foods we eat, the clothes we wear, the buildings we reside in, and the medicine we take. They are allowing us to be able to truly innovate and be resourceful and adaptive to all of the challenges that we face ahead of us. I would argue that they are also extremely fundamental for our mental health, and I personally believe that they have intrinsic value. And I think that something that we need to understand is that we are at a place right now where plants are facing incredible challenges. And I want to talk about ways in which we can have a way forward to face the challenges that we're dealing with today. So, a very alarming number of 45% of our plants globally are at risk of extinction. This is, of course, due to all of the impacts from habitat loss, including deforestation, urbanization, overuse of agriculture, also things like invasive species, pollution, and overexploitation. These are really driving forward all of the crises that we're seeing in the loss of biodiversity. But the biodiversity we face, the loss of biodiversity that we face, is not just about individual species. It's really about the unraveling of entire ecosystems that sustain our life. And that includes, of course, human life. And while we're witnessing this really dramatic decline in biodiversity, I believe that we can face that with a really coordinated effort working together in unison across multiple organizations. That 45% number, often I get asked, well, that's of course because of the incredible diversity in the tropics and all of the issues that we're facing there. But when we look closer to home, here in the United States, we see that 34% of our plant species are at risk of extinction. And something that's really important to understand is this is not about our great-great-grandchildren or our grandchildren's problem. This is our problem today, right now. When we look at plants that have vanished from our ecosystems since colonization, we can see that 40% of the species that once resided in the southeastern United States that were native, our 40% of those were actually lost in the southeast. This is because the Southeast is one of the biodiversity hotspots in the United States, and it's one of the really exciting ways in which our garden gets to participate in trying to actually stop the loss of those species. We also are dealing with our loss of climate change. So climate is no longer a distant issue in the past. It's really a daily reality that is shaping the work that we're doing today. And currently at the Southeastern Center for Conservation, where I work, we are finding that more than half of our projects are actually impacted by climate change. Various serious impacts from the shifting frequency and intensity of storms and shifting weather patterns are reminding us of how vulnerable our ecosystems are, and especially our rare and imperiled plants in the region. And I would say rather than feeling disheartened, we must really work together to use this as a call to action so that we can focus on building community, resilience, and cooperation amongst each other. We have to focus on embracing the different values as we move forward, finding ways to be innovative and to protect plant species together. And one of the solutions to this is botanic gardens. And I see that my slides have, despite the fact that I checked them earlier, are not showing up correctly. So I'm very sorry. You can't read any of the text. So I'm really sorry. I checked, they worked earlier. But this slide is supposed to talk to you about the importance of botanic gardens and how we are truly uniquely equipped to be part of the solution and how we are able to really combine a deep horticulture expertise and, in addition, scientific expertise. So we provide not only the physical structures where we're able to grow these incredibly rare species, but we're also able to communicate the importance of those plants to people in the area. So we are more than just beautiful places that people come to have parties and find a moment of quiet respite. We are places that grow plants, and we also grow awareness. So gardens are able to connect millions of people each year to the wonder of the plant world. And often we're bringing in people for the very first time. And we are storytellers, we're educators, and we're conveners. So we bring people together and we're bridging the gaps that we find in our community. So we bring our scientists together, we bring our policymakers together, various community members, and other partners to be able to participate in conversations around the importance of plants. And we always are focusing on linking plants and the well-being of people. Again, I'm sorry that you don't get to see the pretty slides that I worked on. But one of the definitions that we actually see from Botanical Gardens Conservation International is really focusing on how Botanic Gardens missions should focus on scientific research and also conservation. And these are both paramount to the work that we're doing. And it allows us to play really pivotal roles in being able to combat the loss of biodiversity. And we have more than 3,700 gardens that are part of this incredible Botanical Gardens Conservation International framework. And this really allows us to work in unison together. And so as we look at an integrated approach, when we think about the mission and how we are actually working together to encourage our mission to use that scientific knowledge and our conservation, we can see this beautiful approach of how we focus on this integration together of all of the different aspects that Botanic Gardens can bring. And so this was a wonderful diagram that was created to show representative of how we can work together. So we have our in situ work, and that's really working in the wild where the natural plants are occurring. And then we have our ex situ work, which is our collections and all of the different myriad of kinds of collections from our different germplasm with our seed banks, our cryo and living collections. We also have all of our conservation collections, reference collections, and our display. And I will add that we put in meta collections, which I'm going to talk to you about here at the end. We can also see how research, horticulture, and education tie in and play critical roles for each of these components that we're working together. At the Southeastern Center for Conservation, we developed our workflow around this idea of this integration. And so this is the workflow that we use on our team to be able to really show people and how our work is all tied together. I will say that not a singular spoke of this wheel could be removed and our work be as meaningful as it is. So we focus always first on understanding baseline, what's out there, what's going on. This is really our in situ work, our in the wild work. From there, we go and we seed bank anything we possibly can, because seed banking, of course, is our most economical and space resourceful way to conserve plants. From there, we have our non-orthodox species, or species that can't be traditionally banked. So we then work in our cryo and our in vitro work, making sure that we're able to conserve those species that are not traditionally kept. And we have our living collections in our conservation horticulture team. And these are the people who are really growing the hardest and most unusual plants, plants that have never been in trade before and that are coming into collections for the first time. And something that we focus and our team is making sure that all of that ex situ work, or the work that we're doing out in the garden in our collections, is all genetically sound. We don't want to be making sure that we're just collecting the prettiest plants out there. We want to make sure that we're getting the full genetic representation of what's left in the wild so that we can use that material if we need to in the future. We also then have our conservation ecology, and that's really making sure that we're taking an ecological approach. Because like I talked about, one species is really important. Thinking about an individual is important. But what we want to make sure is that we're conserving the ecosystem. Because if we put one plant back out there without thinking about the system it's connected to, we're going to fail. And so that leads us into all of the great work in our collections being able to help support experimental augmentation and habitat restoration that drives forward that stability of those systems that we're trying to work to conserve. And finally, I cannot say this strongly enough, but the center and core, and if you notice that the background of the slide is, or the background of the circle is also purple, that's because networks and partnerships are fundamental. There is not a more important role that we play as just a member of networks and the partnerships that we're part of. We could not do this alone. We're one small institution trying to make a difference, but it's the power that we work collaboratively and collectively together that allows us to actually do the work in a meaningful way. So I will argue that I will have a very bold argument that there is truly no reason that our scientific knowledge does not allow us to prevent plant extinction at this time. We currently have the tools, we have the technology to prevent plant extinction. What we lack is absolutely not ability or knowledge, it's alignment. So to stop extinction, we need to really shift how our resources are allocated. We need a deeper investment in capacity building, especially in biodiversity hotspots. And most critically, we need to shift our collective priorities. I think that we need to understand that plants are fundamental to Earth, that we cannot continue to have them underrepresented in conservation policy and funding. Currently, plants are at the bottom of the funding bucket. And if we're serious about protecting biodiversity, we really have to recognize that saving plants is not optional, it's absolutely essential. And so this slide would have had a list of all of the incredible cool things that botanic gardens do. And that includes things like ex situ conservation and research and our collections, policy and advocacy, education and outreach. And I am just going to focus today on our ex situ conservation because we're here talking about collections. And so this is terrible. I'm so sorry. But this is basically trying to explain to you what ex situ conservation is. And really it is basically having collections outside of their wild natural spaces. So this is zoos, aquaria, and botanic gardens that have our collections. And we know that right now we have significant number of our plants. We have about 40% of our plants that are currently in, of our threatened species are actually in collections, and around 30% of all of the plants known are in some botanic garden collection. However, that's one singular instance often. That is not enough to save a species. And we also have examples. This is Franklinia here. We have about 100 extinct in the wild species that are in our collections. So it's extremely important and wonderful for us to have these collections, but we need to push forward and move into a new way of actually doing our ex situ conservation to ensure that we're maintaining them, our work for the future generations. And so this slide is going over why and how ex situ conservation collections are used and how they're important. So they serve as a safety net. This really allows us to be able to have, if we do them correctly, to be able to have enough material to work with saving an individual population if we have enough material in collections. We also can make sure that we're preserving enough and broad enough genetic diversity that's actually found in the wild. We can facilitate research and prevent the over-exploitation or over-collection of individual populations through our ex situ collections access. And we can support reintroduction programs where we're actually able to go back into the wild and focus on getting plants augmented or reintroduced. And so I'm going to briefly talk about the current challenges that we have in ex situ conservation and how we're trying to address them. So we have issues regarding prioritization. That 45% number is pretty significant. We need to be able to figure out how to address that in a meaningful way so that our funders and the people who want to make a difference can actually understand what the species they can work on to make a biggest amount of impact on. We also need to do a better job at integrating our ex situ and in situ conservation because saving those individual species in the wild is absolutely at the heart of what we need to do. We have to overcome hurdles because we do have a lot of species that it's quite confusing and we don't know how to germinate yet, and we don't know how to bring them into cultivation. And then ultimately, we have to make sure that we maintain that genetic diversity. So here I'm talking about the first, which is prioritization of our the work that we're doing across the Southeast. So we have done two very large prioritization endeavors where we've been able to focus on, with many, many partners, on creating a gap analysis for the highest priority taxa that are currently in collections. What we were able to find is that 54% of our rare and imperiled plants are actually in collections. So we're doing a really great job already. But what we're finding is only 32% of them actually have any kind of documentation in wild origin. That would be awesome. Thank you. I think you'll enjoy it a little bit more if you can actually read it well. It's one. Mm-hmm. Oh, see, they look prettier. Yep. I'm right here. Awesome. Thank you. All right. There you go. You can read the text. I will learn never to use white text again if that ever could happen. So you can see here that we're able to actually understand what kind of impact we have had with our ex situ collections. This is really important because it means we know how many species we need to focus on. We also were able to create the regional, this is a horrible acronym, or RSGCN, are the first plant regional species of greatest conservation need list. And this is really for us to be able to look at the 15 states in the southeastern US and be able to understand what are the most high and very high plants that we actually have to focus on. This really does allow us to prioritize that funding priorities. When we look at trying to figure out ways in which we can integrate our work better, our ex situ conservation, we need to make sure that we're really complementing all of our in situ efforts. This includes our habitat protection, restoration, and reintroduction. And these are a couple of the ways in which we're trying to use newest technologies to be able to incorporate a better use of our ex situ and in situ, everything from looking at our fungal communities to get the big broader picture. We're able to use detector dogs to be able to find some of our rarest and hardest to find species, doing forest habitat structure composition studies. This allows our horticulturists also to better understand how to grow these species. And finally, doing experimental management to really figure out what we can do best with that in situ work. And then when we look at our biological hurdles, we were finding out of some of the really great work that came out of the North American Orchid Group at the Smithsonian, that making sure we were doing symbiotic germination of our orchids was really important. We were having trouble with some of our outplantings of orchids because we were growing them asymbiotically. And so using the newest innovations and learning from our partners about how to do experimental augmentations with symbiotically grown orchids has really improved our conservation efforts. And when we look at additional biological hurdles, we can see how we're making huge improvements. A lot of this new technology is coming out of the agricultural sector because they are really light years ahead of us in terms of their advancements in research. And so if we utilize these new best techniques and apply them to conservation, it allows us to do more with fewer amounts of dollars. And so we can see things from our cryo to our seed storage longevity, integrating genomics more, and then making sure that we're looking at that whole ecosystem. So preserving our microbiome is also really important. And We also need to think about genetic diversity. This is something that's really near and dear to my heart because I am so concerned about that next step and the integration with in the in situ work. And so everything that we've been focusing on at the garden has been really around focusing on the Center for Plant Conservation's CPC best practices, including maternal line tracking. So what does that mean? It's quite simple, I think, if we think about it. If you look at each of these little black circles, each one of the circles represents a population, and the different colors represent different genotypes that we see in those communities. When we go out and do a collecting event, we're going to collect from different individuals, so different genotypes that we're actually able to collect. When we do bulking of that seed, which is a more traditional, common way that we used to do because it's a lot more time efficient, we would find that we would end up with this pooled population. This is really problematic because it can mean that we can artificially select one particular type of genotype. So you can see this sort of olive individual genotype is the only one that's getting represented in this bulk collection. And that could just be due to the fact that those seeds were better and had stronger likelihood of succeeding. When we do collections by maternal line, so if we go out and we're collecting all of our different individuals and we can then keep them separated, so this darker green and this more olive green, we're going to separate out and keep them separate through the entirety of the storage that we have them in collections. We're able to make sure that we actually can keep each individual genotype represented. This allows us to do germination trials per maternal individual and really helps us to understand when we don't have the funds to do genetics on all of our collections, we can at least estimate that we have a good amount of genetic diversity conserved. So this is really awesome. And let's say we've gone out and our garden has all of these individuals. Great, wonderful. But it's really important that we don't put all of those collections in one place. And this is because there's a lot of different reasons that I'm going to go into that shows why we shouldn't. And what the solution going forward should be is to meta-collect. So this is our greenhouse. And if we kept all of these Sarracenia to ourselves and did not share these out, then if we had a pathogen, if something happened to our collection, we could end up losing a huge amount of genetic diversity, which would be really absolutely against what I think conservation is at the heart of. So what is a meta-collection? A meta-collection is, very simply put, a collection of collections. And so it's a group of institutions working together to collectively prevent extinction and to steward collaboratively their collections of many species. This is really important. And the work that was started by Patrick Griffith at the Montgomery Botanical Center and all of his collaborators really has pushed forward this idea and the work of working together. So we are always trying to, when we have our different meta collections, which is really key for all of the taxa, the imperiled taxa we're working on, we're trying to ensure that we're getting 150 to 200 individuals per population or up to 3 clones per critically endangered species. It's a huge number of plants. And if you think about the resources, that would be way more than any botanical garden could manage alone. And so it's really important that we actually distribute the risks and we distribute the load. So this just sort of shows you the benefits where we're distributing risks, we're maintaining genetic diversity because we can have more institutions. Instead of keeping 150 individuals, my institution can keep 4 and another institution can keep 6. And we also see our reduction in costs because we now are not taking care of 150 plants, we're taking care of 6. We also need redundancy. Redundancy is critical and important because pathogens happen, disease happens, we end up with hurricanes, facilities can end up with impacts from various different storms. So it's really important to have that redundancy. And it also allows us to increase that genetic range and start doing things like looking at climate tolerances. I'm going to give you one quick example of Magnolia ashii that we work on through the Global Conservation Consortium for Magnolia. And this is the rarest magnolia that we have in North America. There are many, many rare magnolias throughout the world, but this is our rarest here in the United States. And we're focusing on ex situ work, research, and also a lot of public engagement. So this is work that's been done collaboratively with the U.S. National Arboretum, and we are incredibly thankful for their collaboration in this. They went out and they surveyed all of the different populations across the panhandle of Florida. And they brought these individuals back, they grew them up, and they also were taking genetic tissue samples while they were out there. And so you can see here, this is just a genetic analysis. This is the admixture analysis in which we're looking, each of the bars represents an individual, the different colors represent different genotypes. And what we were able to see is this genetic diversity across the range. And then as we were distributing this material, so we have 40 different maternal lines that are collected across this range. And we've got 10 different sites that are represented. And at the end, when we've actually distributed this material out, so here are the little seedlings that went out. These are the different gardens across the US, and you can see the colors represent the different genotypes that are represented in these collections. So different gardens can hold different levels of genetic diversity, which really is able to actually allow us to ensure that we are protecting the entirety of that population across the panhandle. This is really exciting work. And it also allows us to test climatic tolerances of some of these species, because obviously the plants are growing down here in the Florida Panhandle. And you can see as we're trialing these species at different gardens, we're able to really test those tolerances. And so in summary, we can actually see huge work that's being done across our network, where we have seed banks and tissue culture facilities. We have large ex situ programs that are occurring across our different botanic gardens. At Center for Plant Conservation, we have more than 780 species that are being collectively, that are being used in these meta collection styles. So it's really important to see how this work is being applied in all of the practices that we're doing today. And so I'm going to leave you with the future of what I think ex situ conservation could be. I see that as we work together towards more meta collections, we can actually conserve more species with fewer dollars. We can really work on developing new implementation for best planning and strategically thinking forward. We can work on doing a better job at experimental translocations and augmentations. I think we need to focus on building capacity in those biodiversity rich countries in which a lot of our rarest of rare species are occurring. We need to be working with our local communities. We've got to get out and work with them, and botanic gardens can be that first step in doing that. We need to do more with our technology, with our drones and our genomics, integrating these and cryopreservation into all of our work. But at the heart of it, it's collaboration. There is absolutely no way we can do this without working together. And so I'm going to leave you today with this idea of how critically important I think ex situ collections are. And I'm going to say something bold in that I really think we're at a crossroads in history. I believe that plant conservation, while I have had many grant terminations, and I know the struggles and stress we're all facing, but I do believe we're also at a golden age for plant conservation. I believe that because the knowledge that is held, the diversity in partners that we are creating right now is greater than it has ever been. I think that we are growing stronger paths forward. And I believe that while our ecosystems are at a more vulnerable place than they've ever been, that the willpower behind the human heart and the willingness to work together is what will actually make us really change the course of history. I have to believe this because I have dedicated my life to plant conservation. And I really believe that we can work on empowering each other and empowering one another so that we can have this great golden age of being able to share knowledge and work together collaboratively. I really don't think that the work should be isolated to small institutions. It's about working together. And so I would ask each of you to join in that. And I like to leave people with ways in which they can help, because I think all of us feel like we need to do something. And so these are some of the ways in which I would recommend that you could help. And there's so many others, and please share those amongst yourselves. So I think just sharing personally the intrinsic value and beauty that you find and see in plants is really important. Talking about plants to people that don't know much about them is actually kind of fun. I think it's also really important that we all try to learn more about the wild plants that are in our area and how we can encourage more native plants in our backyards and avoid over-collecting. I also think it's really important that we are at a time right now where you using your voice is so important. Um, I would recommend there is the open period for public comment for changes to the Endangered Species Act. That's open until May 19th. So please reach out, speak up that our Endangered Species Act, especially for plants, is so critical. Plants would get hit twice if if the change to harm gets impacted, because plants are essential to all of the animals that live on this earth. So plants are fundamental to not only the endangered species that are on that list, but also to all of the animals that are. And I would say support your Botanic Garden and other NGOs that are doing work in conservation efforts. And finally, always be willing to use your voice to advocate for plants. They do not have a voice of their own, and they need us to speak for them. And so I encourage each and every one of you to use that voice and speak about being able to make a difference in plant conservation. And so with that, thank you for dealing with all of my technical challenges, and I really appreciate your attention, and I hope that you learned a little bit about Botanic Gardens. Thank you.
A fantastic talk, Emily. Thank you so much. I have a question online and I have a question of my own, and I'm going to ask them both at the same time because they're related. Charles asks, except for a few rare individuals, there has always been a bit of disconnect between taxonomous and cultivated plants. Many authors have pointed to an alarming decline in crop diversity over the last century. Should botanic gardens be more involved with crops and their wild relatives? And then my question is, one thing I heard you say was that instead of focusing on pretty plants, you want to focus on genetic diversity. So how do you balance what the public wants versus what Botanic Gardens is giving?
Absolutely. And I think that crop wild relatives are so cool, and we should absolutely do more of that work in Botanic Gardens, because what better way to connect people and plants, like the food you eat, and this is what, this various different plant species originated from. So I think that there is a great movement. The U.S. Botanic Garden has been really elevating the importance of crop wild relatives, and I would encourage people to look into that. There's a lot of great resources and more that's being done in that. And then I would say your other question was, yes, pretty plants versus genetically distinct, and like our collections at our garden, where we have literally hundreds and hundreds of the same species. So I think that there is a careful balance. I think you can have both display gardens, which are so critically important because they get people in the door. They give people those beautiful spaces. But I think that's where meta collection really comes in, because you can start to weave in those incredibly important meta collected plants that are part of this greater network, that have data and are able to be really meaningful. And so if you intersperse those into your display gardens, and then we work with partners, even if we're working with non-traditional partners, to help us maintain those genetically diverse meta collections. This is where we've been working with the U.S. Forest Service research stations. We're putting meta collections on their lands. We could work with golf courses as all crazy, idea of partnering with, to actually have them keep some of our trees in meta collection. So I think we need to be really creative. We need to look at new alternatives. We're at this moment where we can't think in the same box we've always thought in. We have to think absolutely broad. Who are our new partners? And I do think that you can also display large groups of species very beautifully. So I think we can also change people's perceptions on what beauty is and different ways to look at beauty. So I am an always optimist that believes that we can start to teach people, and that's the outreach and education and showing people new ways in which we can engage and bring plants into collections in different, more meaningful ways.